Hi, everyone. I just want to say thank you for all the thoughts and prayers for my family situation, but I'm finally back home. So I'm just going to record it. Hopefully, I'll upload it by tonight. Today's Friday. So hopefully, I'll have it uploaded by tonight. That way, you guys can at least go through the lecture before we have an office hours in the upcoming days. So today, we're going over is lectures 44 to 47. And then for next week, we're just going to finish off those last couple of slides for your last exam. I don't know if we're going to have time for a review just because this exam is on a Monday instead of a Thursday. So it kind of screws up everyone's schedule, especially because it's finals weeks as well. But I'll try to set up some little reviews or at least in minimum set up some practice problems so you guys have extra practice for this exam. So going into lecture 44. So lecture 44, we begin talking about eukaryotic transcription and gene regulation. So an important thing to know is that your eukaryotic transcription is very much dependent on the way that a DNA is associated with the histones. So an important term to know is that a nucleosome is a DNA associated with histones. DNA is associated with histones based on the electrostatic interactions and hydrogen bonds that occur between the positively charged histone and negatively charged DNA. So usually what you're going to see when there's an active transcription, we call it a bead on a string because you're going to see just a string, which is your DNA, and a bunch of beads, which is just our RNA polymerases doing its work. So there's two different conformations for the for our DNA when associated with histones. We have our heterochromatin, which is just a condensed chromatin structure, which is not available for transcription. And we have our euchromatin, which is just the opposite. We have an open chromatin structure, which is available for transcription. So how do we open that chromatin up? There's two different ways. We have our chromatin remodeling complex and our histone acetyl transferase. So usually when we're talking about our histone acetyl transferase, I actually put it here on the right. So we have three or two different enzymes that are involved with acetylation. We have our histone acetyltransferase, our HAT, which acetylates the lysine in the histone tail, and we have our histone deacetylase, which removes the acetyl groups from the histone tail. The important thing about the histone tail, because this is the area of the histone which is where all the interaction takes place. So this is where a lot of our regulatory mechanisms are going to be taking place. So as I say right here, when we have acetylation is when we're favoring an open chromatin. So our histone acetyltransferase is actually going to allow for an euchromatin, while a histone deacetylase is actually going to favor a heterochromatin. Our hypermetallation is done by the histone metal transferase, and this metallates the lysine and arginine in those histone tails. And histone metal transferase is actually going to favor heterochromatin. So an important thing that I always try to bring up is how to read a histone code. So you see a histone code example right here is our H2BK5. So H2B is just referring to the histone subunit, and K is the amino acid, which is lysine in this case, and 5 is just the position in the primary sequence of the histone. So what does acetylation do? So acetylation actually neutralizes that positive charge of the lysine in the histone tail. So this in turn reduces the electrostatic interaction that occurs between the DNA and the histone, and it favors the recruitment of a chromatin remodeling complex. So our chromatin remodeling complex, the most popular one that we like to use is our SNF. So this actually alters the chromatin structure by repositioning, unwrapping, and evicting the nucleosome. So it just rearranges the nucleosomes in its most general sense. It rearranges the nucleosome to favor a more open chromatin structure. Right? So talk about DNA metallation. So we talked about histone metallation. So this is now DNA metallation. So DNA metallation occurs in a specific area called our CPG. This is a sequence of the DNA that can be hypermetylated, and it's done by the DNA metal transferase. So these methyl groups actually protrude into the major grooves of our DNA and actually block some transcription factors. This is not always the case, but this is just one example of how DNA metallation can actually inhibit transcription. So we talked about our cis-acting elements when we talked about the prokaryotic transcriptions. For the eukaryotic transcription, it's pretty much similar, except that we have something called our promoter proximal elements, which are located upstream from our promoter site. So this is the areas right here. And we actually have enhancers, which are located distal from the promoter side and the +1 side of transcription. So these are going to become really, really important really soon. So before we have to talk about the DNA binding domains and the activation domains. So this is just a general schematic of what a transcription factor is going to look like. So right now we're going to be focusing on this DNA binding domain. So these are the different ways that we can actually bind to the DNA. We have our helix turn helix, our homeodomain, classical zinc fingers, nuclear receptor zinc fingers, and our dimers, which is our leucine zipper and our helix loop helix. So for a homeodomain, this is a transcription fact, or this is a DNA binding domain that's important for developmental processes. It involves 3 helices, and the third one is the one that's recognizing the base pairings of the DNA. We have our classical zinc finger. This is coordination between 2 cysteines and 2 histidines with a zinc in the middle. And this is used to recognize very unique and distinct DNA sequences. So just going a little bit more in depth about the zinc fingers, usually recognition helixes can have many zinc fingers, and therefore each zinc finger is going to recognize a very specific base of the DNA. And this is pretty important because it improves and increases the specificity that a transcription factor must have for transcription, for them to do what they have to do, either activating transcription or repressing it. So zinc fingers is basically acting very much as a multiple keys in a lock in a sense. So each zinc finger is going to be coordinating with a specific base sequence. So everything needs to be in order before a transcription factor can do what it does. So then we have our nuclear receptor zinc finger. So this actually binds to DNA as a dimer. So both subunits contribute to a recognition helix and a zinc finger that supports the structure. Then we have our leucine zipper, which is just a series of leucines aligned along an alpha helix, and the recognition of a helix are extensions of a leucine. And we have our helix loop helix, which is the extension of the alpha helix from the recognition helix. So these are some of the pictures. So the thing that I want you to guys know for these DNA binomains is just these basic informations. They're not going to be really detailed about it. Zinc finger might be a little bit because zinc finger is pretty important in terms of specificity. But other than that, just understanding and knowing some of these little differences between the DNA binomains will be enough. So I'm just going to skip this question since I'm just recording it. So for lecture 45, we're talking a little bit more in detail about our eukaryotic transcription. So exons are going to be the ones that are going to, so each of our eukaryotic genes have both exons and introns. Our exons are going to be the ones that are going to be included in our mature mRNA, and our introns will be eliminated. So primary transcripts will contain both, but then it has to go through processing where we're going to remove a lot of our introns. So there's 3 very specific RNA polymerases for eukaryotic DNA. We have RNA polymerase 1, which synthesizes rRNA, polymerase 2, which makes mRNA and some other small RNAs, and polymerase 3, which generates tRNA and some other small RNAs. RNA polymerase 2 is basically the same mechanism as prokaryotes, but the difference in eukaryotic RNA polymerase 2 is that it does not identify promoters and it must be recruited by different means. So how do we activate transcription in eukaryotes? Before starting transcription, usually a large protein complex will be established on a cis-acting element of our DNA. And remember, a cis-acting element is any area of the DNA that's going to be having some effect, either repressing or activating. So it's going to be in the DNA. So this is usually the order that goes. We're going to bind up some transcription factors to the promoter sites and the enhancer sites. We're going to recruit some co-activators, then we're going to recruit a mediator, and then we're going to have the assembly of the transcription pre-initiation complex. So we just talked about the DNA binding domain for a transcription factor. Now we're going to look at it as a whole. So eukaryotic activators are going to actually exert a positive gene regulation. So the activation domain right here is where the co-activators can actually bind, and these co-activators will often recruit other components that are going to be important in creating that pre-initiation complex. We can also have eukaryotic enhancers. These are located distally from the promoter sites, as we looked about before, and these often help initiate different levels of transcripts. We can actually even bend the DNA in certain ways to promote transcription through something called our HMG proteins. So now that we have these transcription factors, they're going to actually call in the mediator. Sorry, I need to take a little drink of water. So our mediators are the primary means of communications between our transcription activators in the pre-initiation complex. So many of the subunits of our mediators will actually make contact with the transcription factor activation domain, so this area right here. And it's also going to start making contact with the transcription complex. So this is, as it states, is the mediator between the transcription activators or the transcription factors and the whole pre-initiation complex, and it can bind to multiple activation domain of our transcription factors to make contact with the transcription pre-initiation complex. So this is just the order of how the transcription pre-initiation complex is built upon. So only the highlighted ones are going to be the important ones to know. So our TF2D is going to actually bind to the promoter site. Then our TF2B is going to bind to the DNA and our TVP, which is our transcription binding protein. Then our TF2F and our RNA polymerase 2 will join the complex. And then our TF2E and TF2H will enter the complex in succession. And then TF2H is the one really key important one because it has some distinct functions. It can act as a DNA helicase to generate that transcription bubble, and it acts as a protein kinase, which is going to phosphorylate that RNA polymerase 2 carbon terminal domain to initiate transcription. So right here is just an example of everything we just talked about, but looking at the HO gene activation in yeast. I'm just not going to read it because I think you all will be able to understand exactly what's going on here. This is just an example of putting everything together we talked about in the last 2 lectures. So another important thing is how we can regulate genes for eukaryotes. So we have something called our combinatorial control. This is just how we can coordinate between different transcription factors to give out either a high amount of a certain gene or a low amount or none. So this is just a mix match of transcription factors that will give us different results. We can also have repressors. So these are transcription factors that actually inhibit transcription. So there's different ways they can do that. It can act as a competitive binding to the activator binding site, displacing the activator. It can bind to the activator to prevent the interaction with mediators. It can alter the assembly of the pre-initiation complex, and it can provide a docking site for histone deacetylase, right? Remember, histone deacetylase is going to remove the acetyl groups from our histone, which is going to prevent the DNA from being in the U chromosome structure. So methylation is also pretty important too. So gene silencing can occur through methylation. So this is involved. So once we methylate certain histones, they're actually going to become really associated with heterochromatin. So a certain histone like this one right here, which is our third histone here, once it's metylated, we can actually look at it. That's really associated with heterochromatin. So once we metylate it from its original to this new version, this is done through a histone metal transferase, so we talked about before. And the interesting thing here as well is that once we methylate it, it can actually become a docking site for other heterochromatin proteins. And then we can actually form a dimerization of the heterochrom proteins, causing a formation of a more compact chromatin. So I know that was just a lot of words. So basically saying that once we methylated, we can actually use it as a docking site for other heterochromatin proteins, which is then going to start almost like a wave and make all the nucleosomes around it heterochromatin to make it into a compact chromatin. For lecture 46, right? So we just finished our mRNA because remember, eukaryotic transcription is very similar to prokaryotic transcription. So now that we have our mRNA, we need to go through RNA processing. So this is just some of the steps that we're going to look through. So we're going to have a 5' capping, a the cleavage of a poly A site, polyadenation, and then RNA splicing. So before we talk about this, transcription usually occurs in sometimes called transcription factories, which is just transcription clusters. So these are areas of our cells that have multiple RNA polymerase 2s, and these are areas that transcription is constantly occurring. Usually they're just transcribing the same class of genes, and RNA processing occurring in the same site as well. So we're looking at transcription right at the end. So the important thing to know is looking at that carbon terminal domain, our RNA polymerase 2. So this is the repeat of amino acids that we see. So serine, as we know, has a hydroxy group and can be phosphorylated by a protein kinase. So phosphorylation is necessary for the end of transcription. So this allows promoter escape. It also provides a docking site for our capping enzymes, cap binding complex, our cleavage complex, and our RNA splicing factor. So that carbon terminal domain of our RNA polymerase 2 is really important in initiating and terminating. So this is just, as we're seeing right here, As we phosphorylated, this is the area of the carbon terminal domain, and we can phosphorylate it to start ending that transcription. So the first thing that comes off is our assembly of our 5' cap in our mRNA. So the 5' cap is assembled at the 5' end of our mRNA by the capping enzyme, and this is important for protection and the initiation of translation. So I know that this is a horrible looking graph, but what I would know for this exam is know that it's a 5 to 5' triphosphate linkage. So there's 3 phosphates between the cap 0 and cap 1. Understand that what at least the first 3 enzymes do, and understand what cap 0 means. And cap 0 is just meaning that it's essentially a 5' cap. It's our 7-metal guanosine. So our 5' cap is made, right? So now we're going to look at a transcription termination. Sorry, I kind of confused you guys with this one, but this is just showing the carbon terminal domain and where we can actually phosphorylate it. So our transcription termination, this is the signal that we're going to see in our mRNA, and this is after we see the signal, the transcript is still extended to beyond the termination. So termination factors will recognize that sequence and perform the cleavage for the transcript of the from the RNA polymerase 2. And our 3' end then undergoes a polyadenylation by poly-A polymerase, so adding a bunch of adenines at the end, right? So now the most important thing for RNA processing is our spliceosome. So this is a nuclear complex that's important for removing introns and ligating exons. So many of our spliceosome, there's a lot of proteins involved, but the most important ones are our snRNPs and our snRNAs. And we have 5 different snRNPs that have very specific function in our spliceosome. So we have our U1, U2, U4, U5, and U6. Okay, so you can read their function here, but we're going to go over what they exactly do. But before we start, we have to know that in our intron, which is just the blue area in this graph, there's a rule that governs RNA splicing. So we have our GU-AG rule. So our GU is going to be located near the 5' splice site, and our AG is going to be located in our 3' splice site. And then we have a branch point A, which is closer to AG, which just has an adenine right there. So how does this work? So our U1 sRNP is going to be recruited to the 5' splice site, and our U2 is going to be recruited to our branch site A. Our U5, U4, U6 are going to bind to the complex to complete that spliceosome assembly. And then our U1 and U4 is going to leave, leaving U5, U6, and U2, which is our active conformation, right? So let's say if someone asks, what is the active conformation of a spliceosome? Does it involve U1 and U4? No. U1 and U4 come in at first, but they're mostly there to stabilize the structure. But our catalytically active spliceosome is only involved with U2, U5, and U6. So looking a little closer to our U1 and U2 branch points, right? Our 5' splice site, our branch site A, you're gonna see that our U1 has a specific area right here, right? And our 5' splice site that's gonna be able to bind. You see our GU right here, equivalent right here. But we also have this side, which is our pseudouridine. So our side just allows for greater variability in our base pairing. Okay, so even if there is different base pairs right here than is normally encountered, our pseudouridine is still going to be able to bind it to some extent. And our U2 is going to branch to our branch site A, and it's going to have similar sequences as well. This hydroxy group right here is going to be important, so just wait up a little bit. So our U2, U5 base pair to each other, aligning the branch site with the 5' splice site for their first reaction. And you're gonna see that we're gonna create this little lariat right here, right? It's like a little lasso. So then we're gonna rearrange the 5' and 3' splice site for the second reaction, and thus it's gonna have a 2-step trans esterification where the intron is gonna be removed as a lariat, and then we're gonna be able to ligate that exons together. So it's a lot easier to look at it in a more chemistry sense. I know this like the picture right here is a little bit off, but if you're able to look at Dr. Duma's lectures, it makes a little bit more sense. So we see the intron right here. So in our branch point A, remember that we have that hydroxy group. So the hydroxy group is actually going to attack a phosphoryl group that's located right here. So then we're going to form that lariat that we see right here, right? And then our OH group from our exon is going to attack the second phosphoryl that's at the end of the second exon, and is going to connect each other while this whole section right here is removed. So I know it's a little bit off, but just look at the graph and it'll be a lot easier to understand it that way through the chemistry instead of just looking through the graph right here. So an important thing for RNA processing is that we can have alternative splicing. So these are mechanisms for tissue-specific productions of different mRNAs. So we can actually select the exons that we want, and we can rearrange them in different ways. So I've never seen they ask in detail about these alternative splicings, like what is our what's going to happen in an intron retention, or what's going to happen in an alternative promoter and first exon alternative splicing. But I just take a look at them, maybe understand a little bit, like just understanding the general mechanism, or just understanding what alternative splicing is more than enough in my opinion. But I just look at this graph a little bit just to see, I'm I never want to say exactly if this something's going to be in the exam or not, but I'm like 90% sure that they're not going to be really specific about this, but it's always good to, at least check it a little bit. You never know. So how do we select which alternative splicing we're going to go towards? So the selection of exons is dependent on the proteins either binding at an enhancer or silencer sequence. So an SR can bind either in the exonic splicing enhancer, or an HNRNP can bind to an exonic splicing silencer, and that affects what type of splicing we're going to see. So we can also break apart mRNA. So this is a type of RNA processing. So we can do it by deadenylation, so removing those poly A tails, decapping, removing that 5' cap, or we can cleave it by an endonuclease. We also have an exosome, which is just a multi-enzyme complex in the cytoplasm, which reduces mRNAs back into its nucleotides. So now going to my favorite part of this fourth exam, which is translation. So before we start, we have to look at what a codon is. So a codon is just a series of 3 bases that specifies a specific amino acid. We have a start and codon. We have a 3 stop codons, and a coding sequence is continuous, and it's non-overlapping. Because if we did have overlapping, we'd have, so this is the original amino acid that's being coded. If we had overlapping, we'd have completely different amino acids being coded. So a genetic code is degenerate, and in other words, it's just redundant. So we have multiple codons that code for the same amino acid. There's 4 amino acids that you have, or 4 codons, I mean, that you have to know. We have our AUG, which is our start codon, our UAA, UAG, and UGA, which is our stop codons. We also have 4 different mutations that we have to know. Our nonsense mutation, which introduces a stop codon, our missense mutation, which replaces an amino acid with another, our silent mutation, which just does not alter the encoded protein, and our frame shift mutation, which is frame shifts are the worst ones. So frame shift mutation is caused by an insertion or deletion of a nucleotide. So if we insert it, you see that we completely shift our open reading of our codons, which means different amino acids are going to be coded. The same thing happens if we delete it. But the interesting thing is that if we change by either adding 3 bases or deleting 3 bases, we're actually not going to have a frame shift mutation, because technically there's only going to be 1 extra amino acid, or we're going to lose 1 amino acid, but all the other amino acids are going to be perfectly fine. So our tRNA is our best friend for translation. So they're the ones that are going to be delivering the amino acids to the ribosome. So the amino acids are going to be bound to the 3' end of our tRNA. And we have this anticodon triplet, which is going to base pair with a codon in an anti-parallel orientation. So the primary determinants of an amino acid selections are the base 1 and 2, right? Remember, our codon has 3 bases. So base 1 and 2 are the most important bases for our amino acids. And our third base is considered a wobble base. So a wobble base is the third base of our codon. And a wobble base just is a base that doesn't really determine what amino acid it becomes. It's present there that we hypothesize that it's used to prevent mutations. So you're gonna see, like, for example, for isoleucine right here, the first 2 are determining isoleucine, but the third base can be, for the most part, any of the other 4 bases, and they'll still give you the same amino acid. So the wobble base can be practically anything. So now looking at how we charge up a tRNA. So the tertiary structure of our tRNA is an L shape due to the D and T twisting on itself, and this is important for the positioning in our tRNA synthetase. So we have something called our amino acyl tRNA synthetase. So each amino acid has their own synthetase, and they use ATP to drive this reaction, where we're actually putting in our amino acids and connecting it with our tRNAs to form a charged tRNA. So our anticodon sequence and other sites in our tRNA are going to contribute to the selection of the right amino acid to each tRNA, right? Because we want to make sure that the tRNA that has the anticodon specific for a certain amino acid gets the right amino acid, right? So you're going to see in this diagram right here that we have a synthetase. We use ATP with our amino acid to form our valine phosphate right here. In this case, we're talking about valine. And then our tRNA that's associated with valine will come in, and then we're actually going to allow the valine to get connected to the 3' end of our tRNA. So our ribosome is also the big important person in our translation. So we have our prokaryotes have a 70S ribosome, our eukaryotes have an 80S. Our mitochondria also have a ribosome, but they're a 55S. And our ribosome has 3 very specific binding sites, our A, P, and E sites. So remember that prokaryotes actually have are polycistronic, which means they have multiple areas that can be translated in a single operon. So this is where we're going to see polyosomes, which are multiple ribosomes bound to a single mRNA. These are like really pretty images. So you see all these little circles, dark figures right there are going to be the ribosomes in one specific or in one single mRNA. So yeah. So we're just gonna go through the basics of initiation of our translation, and then for the next week, we're gonna go through elongation, right? So our initiation factors we have 3 of them, IF1 and IF3 and IF2. So our IF1 is gonna bind to the A site, and our IF3 is actually gonna bind to the top of our E site. And these are gonna be preventing both the tRNA binding to the A site, and our IF3 is gonna be preventing the 50S subunit from binding it, right? Because remember, our ribosome has 2 different subunits. We have our small subunit and our large subunit. Our 50S in this case is actually our large one. So we're binding our initiation factors to the small subunit. So our mRNA is actually going to bind to the small subunit by base pairing with the 16S rRNA in the Shine-Dalgarno sequence. And as a result, our AUG, which is our start codon, is going to be positioned specifically at their P site. So this, in this case, if you haven't caught on, we're looking at our prokaryotic translation. That's why we have our 50S as our large subunit and our 30S as our small subunit. And we have our Shine-Dalgarno sequence, which we do not have in eukaryotes. So our Shine-Dalgarno sequence, sorry, is what we actually call our ribosome binding sequence. So this is a specific site on our mRNA where the ribosome knows where to bind to initiate translation. So this is just specific area. So as soon as we know the ribosome is able to recognize the Shine-Dalgarno sequence using this 16S rRNA in our ribosome, we're going to be able to position AUG perfectly at the P site. So the start codon for prokaryotes is a little bit different from eukaryotes. So for prokaryotes, we're actually coding for N-formyl methionine. For eukaryotes, we're just going to code for methionine. So once we put all these initiation factors together and we have the AUG specifically at the P site, our IF2 with bound to GTP is going to bind to our P site. I remember IF2 GTP is going to, sorry, our IF2 GTP is going to bring in our F-formyl methionine tRNA and escort it to the P site. And we're going to position that tRNA specifically at the P site. Our IF2 is then going to hydrolyze this GTP, which is going to result in dissociation of all the other initiation factors. And once IF1 and IF3 are leaving, the 50S subunit, so our large subunit, is going to be able to bind to our small subunit. And now we have our translation initiation complex ready. And as we look at right here, our A side is going to be vacant. Our P side is going to have our formyl methionine tRNA bound to it, and our E site is going to be vacant. Right? So this is it for now. I apologize for this Wednesday for not being able to teach. But let's start. I hope you guys are starting to study for your exam, right? Because this final exam is coming really quick. So for next week, we're just gonna finish off all the lectures, and hopefully for the weekend before your exam, I can set up something for a little review. But have a good night or have a good day whenever you guys see this, and start studying. Okay, you guys got this. You're at the final stretch.